All right, hello everyone. So welcome back to another lecture. So in this lecture, we're going to be looking at skeletal and smooth muscle, focusing primarily on skeletal muscle, and then in particular, we'll look at some anatomical features of skeletal muscle, and then we will progress into looking at the physiology of skeletal muscle in terms of how does it contract. So we'll look at the actual events of a contraction that take place um, along a skeletal muscle cell. All right. So to begin, we have to understand some important anatomy about our muscle. So our muscle is considered, or a skeletal muscle itself is considered an organ that's made up of different tissues and different cells. And so we have the muscle itself, but then associated with the muscle, we have lots of different blood vessels. We have other pieces of connective tissue that are intermingled in and around a particular muscle and a particular organ. We also have nerves and lymphatics as well that supply a muscle with its nerve supply, blood drainage, and lymphatic drainage as well. So our muscle itself. So if you were to go into your arm and pull out a muscle, what you would pull out is you would pull out the muscle itself, and then you'd have what are called fascicles, right? And fascicles are essentially groups or kind of bands of individual muscle cells or individual muscle fibers, all right? And so if you think of like a pencil or think of a straw. So imagine you have a bunch of different pencils. So say you have 10 pencils, and then you wrap a rubber band around those pencils and it keeps them all together. That entire structure, that entire thing is the fascicle, and each individual pencil is one individual muscle fiber, right? So here you see one entire fascicle that has been kind of pulled out, and then out of that fascicle, we pulled out one individual muscle fiber, right? So you'll notice that we're calling this a muscle fiber and not a muscle cell. It is still a cell, but the cell is fibrous in appearance and fibrous in shape and kind of cylindrical. And so we generally refer to it as a muscle fiber, and don't always specifically refer to it as a cell. But you're welcome to call it a cell. You're welcome to call it a fiber. So when we look at a muscle, we see this kind of hierarchy, right? And we just kind of alluded to the hierarchy. So you have a muscle itself, you pull that out, you have a fascicle. You pull out from the fascicle, you have a muscle fiber, and then you pull out from that and you get even smaller components called myofibrils and myofilaments that we'll look at in just a second, right? So surrounding our muscle, or on the outside of our muscle rather, we have a band of connective tissue known as a tendon, all right? And so a tendon is a region of dense, regular connective tissue. So if you're unsure of the different connective tissue types, go back and watch chapter 3 and review the chapter 3 lecture that's talking about histology. And you'll recall that dense regular connective tissue is made up of individual collagen fibers that stretch and connect a muscle to a bone. So here we have a muscle, and here we have a bone, and we're joining those two together via tendon, right? Ligaments are also made of dense regular connective tissue as well, right? So we also have bands of fibrous connective tissue that wrap around our muscle, and one of them is called the epimysium, right? So you'll see the term myo and mysium several times. That's referring to muscle, and the term epi here is referring to on top of. All right. So the term epi on top, mysium muscle. This is on top of our muscle, right? So it is a connective tissue sheath that wraps around the entire muscle itself. You then pull out a fascicle, and you get this other region known as the perimysium, and then you pull out a muscle fiber, and around it you get a region known as the endomysium. So you have both of these here. You have the perimysium and you have the endomysium, and kind of embedded within these connective tissue sheaths and these connective tissue layers are blood vessels and nerves and lymphatics that are supplying the muscle with those different things. So a nervous supply or a blood supply and a blood drainage, right? So if you took out a muscle out of your arm or your leg or your neck or something, you wouldn't just have a muscle, you'd have lots of other accessory structures that are associated with that muscle, all right? So here you can see these layers again. So you see the epimysium, epi meaning on top, perimysium, and again, epimysium around the whole muscle. Peri, in this case, peri is meaning around. And so perimysium wraps around a fascicle. So here's a muscle fascicle wrapping around it. You have your perimysium. So you see perimysium wrapping around here. You pull out an individual muscle fiber, and you have the endomysium, endo meaning within. And so this is the deepest connective tissue sheath or deepest connective tissue layer that wraps around our muscles. So here you have an individual muscle fiber, and around it you'd have the endomysium. Now, you also have another layer that you see down here in this bottom image that we'll look at in a little while called the sarcolemma, all right? And so this brings up another point. The term sarco, you'll see the term sarco a lot in this lecture, and if you read about skeletal muscles and muscle contraction. So you have sarcolemma, sarcomere, sarcoplasm, sarcoplasmic reticulum. All these terms are referring to muscle, all right? So the sarcomere, as we'll see in a little while, is the structural and functional unit of skeletal muscle. And so anytime you see that term sarco, you should begin to think muscle, all right? But the sarcolemma and the endomysium, those are different, okay? They're not the same thing, all right? And we'll go into more detail about the sarcolemma in a little while, okay? So if we look at this hierarchy, we can see a number of things. So we can see these connective tissue sheaths that surround a muscle fascicle, surround the individual muscle cell or muscle fiber. We have nerves, particularly motor neurons. And so as we get into future lectures and we talk about the nervous system in more detail, we'll look at what are called motor neurons and sensory neurons. Sensory neurons bring information back to the brain and spinal cord. Motor neurons take information out from the brain and spinal cord and take it out to some effector. And in this case, the effector is our muscle, all right, our skeletal muscle. You also have blood vessels, so you have arteries and veins that are supplying the muscle fibers with oxygenated blood and draining venous blood away. And depending on the metabolic activity of a muscle cell, it will have more or less arterial blood being supplied to it. And then, of course, we have the muscle fibers themselves, right? So the individual muscle cells. So when we look at these muscle cells, we have, there's different features, there's different characteristics of them, right? So if you recall from histology, whenever we looked at our muscle tissue, which was one of our 4 main tissue types, we said that skeletal muscle was multinucleated, okay? So all that's just a term meaning we have multiple nuclei for one cell, right? So some cells are mononucleated, some are binucleated, some may be multinucleated. And so the way we get this multinucleated cell is you take individual smaller cells during development called myoblasts. So the term myo is referring to muscle, blast. Any cell that ends in blast typically is referring to the building cell, the cell that contributes to the building of something else. And so myoblasts are the cells that contribute to the building of muscle fibers or muscle cells. Each of these myoblasts are mononucleated, but when you have them fused, you get a larger functional cell with many nuclei, right? So you can see all of these, just one nucleus per myoblast. You then begin to fuse these together, so you have 2, and then these 2 fuse, and now you have several in one individual fiber, right? And if you recall from histology, again, these nuclei are often pushed to one side of the cell or the other. So they're usually not in the middle, but they're pushed off to the side near the sarcolemma. And this is because inside the cell, inside the muscle fiber, we have lots of different things. We have lots of organelles, lots of proteins that we will get into talking about in just a little while, okay? So if we take a look inside the muscle fiber, we begin to see different things, all right? So one thing we see is something known as sarcoplasm. Sarcoplasm is essentially cytoplasm, but it's in the muscle cell in particular, so you call it sarcoplasm, right? So again, here's this prefix sarco, okay? You also have a plasma membrane that surrounds our muscle cell or muscle fiber, and this is called the sarcolemma, all right? And it's essentially the same thing as the plasma membrane that surrounds a lot of our other cells, but again, it's surrounding specifically a muscle fiber, a muscle cell, so you call it sarcolemma, okay? It's made up of the phospholipids just like other plasma membranes are. Now, there are areas, and this is what's kind of unique about skeletal muscle cells in terms of their plasma membrane. There are areas where the sarcolemma dives down and extends and it invaginates into the interior of the cell. And this is known as a transverse tubule, sometimes just referred to as a T-tubule, all right? As we'll see later, we won't go into the detail now, but essentially what the T-tubule does is it allows the nerve impulse, the action potential, to reach the inside of the cell, all right? So in order for our cells to contract, our muscle cells to contract, and overall our muscle to contract, you have to get the nerve impulse into the inside of the cell, and the way you accomplish that is via these transverse or T-tubules, all right? Now, as the impulse travels down this T-tubule, it passes by what are called voltage-gated calcium channels that open and ultimately release large quantities of calcium into the sarcoplasm, right? So if we zoom back out and we take a look at the sarcolemma, is made up of phospholipids, so you can see this here, and it has a membrane potential. The muscle cell itself has a membrane potential, which is essentially the same thing as what we talked about with the neuron. So there is a difference in the voltage on one side of the cell versus the other, right? So this is the intracellular environment, this is the extracellular environment. You see that it is more negative on the inside than on the outside. So outside is more positive, inside is more negative. So it has a polarity. These different membrane or these different voltages are separated by the sarcolemma, which again is a phospholipid bilayer, just like our other cells. And in this image in particular, you can see that T-tubule, and you can also see some of the voltage-gated channels as well, all right? So here is the T-tubule. You can see how it invaginates down, and the reason it's T or transverse is because it forms a T because it is perpendicular to the orientation of the sarcolemma itself. So here's the sarcolemma. It runs this way. It runs horizontally in this case. Transverse tubules running vertically. So they form a T, or in other words, they're oriented perpendicular to one another. So as the impulse travels down, you see that it can pass by these voltage-gated calcium channels, open that up, and these yellow dots that you see in here, this is the calcium in this structure called the sarcoplasmic reticulum that we'll look at next. So the sarcoplasmic reticulum, this word should sound somewhat familiar to you because we talked about something called the endoplasmic reticulum, all right? So this is sarcoplasmic reticulum because it's in the muscle, it's sarco. And this is essentially a modified, smooth endoplasmic reticulum, all right? In the muscle cell, this is specialized specifically to hold and to concentrate large quantities of calcium ions, right? So all in this yellow tissue here, all of this in here, all of this is the sarcoplasmic reticulum, and these are the T-tubules. And so as the impulse travels down the T-tubule, it passes by the sarcoplasmic reticulum, causes those channels to open, the calcium channels that are voltage gated, and allows large quantities of calcium to rush out of the cell, right? So if we look here, we can see this hierarchy that we've mentioned a few times. So muscle, muscle fascicle, muscle fiber, myofibril, and myofilament. So what we'll do is we will spend most of our time down at the level of the myofibril and the myofilament level. So this area here and here. So it's this structure here and these small proteins that you see here. So these myofilaments, we'll go into more detail in a few slides here, but these are the proteins, and these are what are actually moving and sliding past each other that produce the contraction that we see on a very gross scale, all right? So these myofilaments make up this large structure called a myofibril. The myofibril is lots of myofibrils are packed into an individual muscle fiber, and then the fibers are grouped into a fascicle, and the fascicles are grouped into an entire muscle. All right, so answer this question here, and then we will stop with part one and move on. So skeletal muscle fibers are what? A, B, C, or D? Are they involuntarily stimulated, multinucleated, non-striated, or are they branched? So pause the video, take a second to try to answer it, and I will move on. So the correct answer is B, right? So our skeletal muscle fibers are voluntarily stimulated, right? We can consciously control and choose to contract them via the innervation via motor neuron. They are multinucleated, as we said. They are striated. So these myofilaments that we see here, this actin and myosin here, that's what contributes to the striated nature of skeletal muscle. So non-striated, that is incorrect, and they are not branched. So we said earlier that they are called muscle fibers. They're cylindrical. Cardiac muscle tends to be branched. Smooth muscle tends to be kind of spindle shape or tapered, and then our skeletal muscle fibers are very cylindrical and very fibrous, right? So we will stop here with part one, and we will pick up with part two, talking about our myofilaments.